Good morning, beta. So quickly answer me through poll. My screen is visible and my audio is clear. Okay. So today we are going to discuss respiration in plants. Can anybody tell me what do you mean by respiration? As you all know that all living organisms, they need energy for carrying out the daily life activities like absorption, transport, movement, breathing, reproduction. Very good, beta. So we all need energy. Right now, I am teaching to you. Okay. I'm teaching like I need energy for that. Okay. Every process need energy. So when we eat food, there's a lot, it consists of like carbohydrate, proteins, right? So the molecules, these larger molecules, they are the macromolecules because they are large in size. So these macromolecules, they are broken down into simpler molecules. And when these molecules are broken down into simpler molecules, the energy is released. Okay, energy is released. And this energy is trapped in the form of ATP synthesis. This energy is trapped in the form of ATP. Okay. This is what we call cellular respiration because this is taking place within the cell, this process. And the compounds that are oxidized during this process are known as a respiratory substrate. It could be carbohydrate, it could be proteins, and it could be fats. Okay, so during oxidation within the cell, all the energy contained in the respiratory substrate is not released free into the cell or in a single step. You know that glycolysis is a 10-step reaction. Okay, it's a 10-step are taking place in the glycolysis pathway. Okay, so when the glucose is broken down into the pyruvate molecule or when the complete oxidation of glucose is taking place, in that case, the glucose is converted to a simpler compounds like carbon dioxide or water, which we call it as a complete oxidation. In this, the energy is not releasing in just one step. The energy, it is releasing in several steps. There are several steps are taking place in this respiratory pathway. So that the energy which is releasing can be trapped in the form of ATP, which is the energy currency of the cell. And the carbon skeleton, which are produced during this respiration, they are used as the precursor for the biosynthesis of other molecule. That is why we also call respiratory pathway as amphibolic pathway, because there's both catabolism and anabolism will going to take place. Next, we are going to discuss is glycolysis. Glycolysis, as the name suggests, glyco is for carbohydrate, sugar, and lysis is breakdown. Okay, this pathway is given by Embedden, Mayorov, and Parnas. That is why we call it as EMP pathway. And glycolysis takes place in cytoplasm. It takes place in cytoplasm. In plants, you remember that the synthesized sugar is glucose, the translocating sugar is sucrose, and the storage sugar is starch. Okay? So glucose is derived from the sucrose, or it can be derived from the storage carbohydrate, that is starch. As you know, the sucrose is a disaccharide. It is a non-reducing sugar. Okay, so being glucose a disaccharide, The sucrose is a disaccharide, so it consists of two units. One is a glucose, which is an aldose sugar, and the second one is a fructose, which is a ketose sugar. So the enzyme which catalyzes this reaction is the invertase, which convert the sucrose into fructose and glucose. And then these two monosaccharides will enter the glycolytic pathway. We are going to discuss the glycolytic pathway in detail because it's very important. So glycolytic pathway consists of two phases. One is the preparatory phase and the second one is the payoff phase. Okay, preparatory phase in which the ATP are utilized, and payoff phase in which the ATP synthesis will takes place. So first step is the phosphorylation step. As you can see, the glucose is converted to glucose 6-phosphate. At 6 position, the phosphate group is going to attached, and the enzyme which is involved in this reaction is hexokinase. Okay, and this enzyme requires ATP. Okay, ATP investment is taking place in this process. First, ATP is utilized here, where the glucose is converted to glucose 6-phosphate. And remember, it is an irreversible reaction. This step, first step, is irreversible, and ATP is utilized in this step. Enzyme involved is hexokinase, which convert the glucose to glucose 6-phosphate. Now this glucose 6-phosphate is converted to fructose 6-phosphate. So the aldose sugar, I told you glucose is the aldose sugar. So glucose is converted to fructose. Fructose is a ketose sugar. So isomerization is taking place. The second step is the isomerization step, and the enzyme involved is phosphoglucoisomerase. Remember the name of the enzyme. And this step is a reversible step. Okay. Now this fructose 6-phosphate is converted into fructose 1,6-bisphosphate. That means ATP is utilized, and the phosphate group from the ATP is attached at one position of the fructose 6-phosphate. Now the fructose 6-phosphate has become fructose 1,6-bisphosphate. Okay, remember this point. And this step is also irreversible like the first step. And the enzyme involved is phosphofructokinase. Remember the name of the enzyme. Now this fructose 1,6-bisphosphate undergo cleavage, and the enzyme involved is aldolase. Aldolase cleave the fructose 1,6-bisphosphate into two compounds. Number one is a glyceraldehyde 3-phosphate, and number two is dihydroxyacetone phosphate. Okay, and this is a reversible step. Now what happened? This dihydroxyacetone phosphate is converted back to glyceraldehyde 3-phosphate. This is also isomerization step. And the enzyme involved in this reaction is triose phosphate isomerase. Okay, so the final product we are getting of the pre-preparatory phase is glyceraldehyde 3-phosphate. Now we are going to continue with the payoff phase. Okay, we have glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is now converted to 1,3-bisphosphoglycerate. In this step, enzyme involved is glyceraldehyde 3-phosphate dehydrogenase. And in this process, NAD is reduced to NADH. Very important. Please remember these steps where NADH is forming, the step where ATP is utilized, the step where ATP formation is taking place. Remember these steps and the name of the enzyme involved in each step because these are very important. Now this 1,3-bisphosphoglycerate will convert it into 3-phosphoglycerate, and the enzyme involved is phosphoglycerate kinase. Now ADP will form the ATP. This ADP You can see this 1,3-bis phosphoglycerate. It has bis. Bis means 2. It has 2 phosphate group. Okay. Now the 1 phosphate group is taken by ADP and ADP is converted to ATP. So ATP is released in this step. And remember, this is a substrate level phosphorylation. Phosphorylation could be of two types. It could be a photophosphorylation or substrate level phosphorylation. Photophosphorylation, which we studied in photosynthesis, where the ATP formation is taking place in the presence of light. Here it is substrate level phosphorylation, where the substrate is acting as a donor for the ATP synthesis. See, there's 1,3-bis phosphoglycerate. It has 2 phosphate group. It has donated 1 phosphate group to ADP, and ADP will convert into ATP. So it is a substrate level phosphorylation. Remember this point, that the step where the substrate level phosphorylation is taking place in glycolysis. Remember this step. And the enzyme involved is phosphoglycerate kinase. Now this 3-phosphoglycerate is converted to 2-phosphoglycerate. Enzyme involved is phosphoglycerate mutase. Now this 2-phosphoglycerate is converted to phosphoenol pyruvate, and the enzyme involved is anolase. Okay, and in this process water is released. Okay, now this phosphoenol pyruvate is converted into pyruvate, which is the end product of the glycolysis. Remember, the end product of glycolysis is pyruvate, and 1 glucose molecule forms 2 molecules of pyruvate. And the enzyme which catalyzes the reaction of when phosphoenol pyruvate is converted to pyruvate, the enzyme which catalyzes this reaction is pyruvate kinase. And here also the ATP formation is taking place. And this ATP formation is also through the substrate level phosphorylation. And this step, it is also an irreversible step. Remember all these points. It is very important that even the NCERT has mentioned the steps where NADH formation is taking place. This step, the step where ATP formation is taking place, the step number 7, step number 10, right? And the step where the ATP utilization is taking place, that is in the pre-preparatory phase. These steps are very important. Please remember these steps. And glycolysis is a 10-step reaction. Next slide shows the table. We have mentioned the enzymes of some enzyme of glycolytic pathway like phosphofructokinase and pyruvate kinase, and their activator and their inhibitor. Like phosphofructokinase, activator is fructose 2,6-bisphosphate, fructose 6-phosphate, and AMP. Activator are those which increase the enzyme activity. Inhibitor are those which suppress or decrease the enzyme activity. So inhibitor of phosphofructokinase is citrate and ATP. Similarly, for pyruvate kinase, the activator is a fructose 1,6-bisphosphate and AMP. And inhibitor is acetyl-CoA and ATP. Remember, the ATP is the inhibitor for this phosphofructokinase and pyruvate kinase, but AMP is the activator. The end product of the 10-step reaction is 2 ATP are forming, 2 pyruvate are forming, and 2 NADH are forming. Generally, in glycolysis, there are 4 ATP are forming. Okay, but 2 we minus 2 because the 2 are utilized in the pre-preparatory phase. Remember, that is why the end product contains only 2 ATP and 2 NADH are formed and 2 pyruvate molecules are formed. And the fate of this pyruvate molecule, if the oxygen is present, it can undergo complete oxidation. If oxygen is absent, can undergo fermentation. Remember, large amount of energy will be released when there's a complete oxidation, because this pyruvate will move to mitochondria where acetyl-CoA is formed. Acetyl-CoA will then enters into the Krebs cycle in the matrix of mitochondria. Then Krebs cycle will produce NADH and FADH2 that will move to the ETS, electron transport chain, and the ATP. A large amount of ATP will be synthesized over there. But if fermentation is taking place, that means there's insufficient oxygen or there's absence of oxygen, then fermentation will take place. And the amount of energy in fermentation is very low, even less than 7%. So fermentation takes place in the absence of oxygen first. Final electron acceptor is the organic compound. In aerobic respiration, when the complete oxidation takes place, in aerobic respiration, the final electron acceptor is oxygen. Remember, final electron acceptor is oxygen, but in fermentation, the final electron acceptor is organic compound. Fermentation is of two types. One is alcoholic fermentation, second one is lactic acid fermentation. In alcoholic fermentation, the pyruvate first convert into acetaldehyde. Enzyme which catalyzes this reaction is pyruvate decarboxylase. Okay, and in this process, CO2 will be released because it's a decarboxylation process. Decarboxylation means that CO2 is going to release. Now this 2-acetaldehyde will convert it to 2 molecules of ethanol. Enzyme which catalyzes this reaction is alcohol dehydrogenase. Okay, and this step utilizes NADH. The NADH which is formed during the glycolytic pathway. Okay, it is utilized in the fermentation. And fermentation takes place when there's absence of oxygen. Now is lactic acid fermentation. In lactic acid fermentation, pyruvate gets converted into lactate, and the enzyme which catalyzes this reaction is lactate dehydrogenase. In this process, no release of CO2. Okay, CO2 is not going to release. So the only process where the CO2 is released in fermentation is the alcoholic fermentation. In alcoholic fermentation, you can see there is a release of CO2, but in lactic acid fermentation, when the pyruvate is converted to lactate, there's no release of CO2. Only NADH will be utilized. And the enzyme which catalyzes this reaction is lactate dehydrogenase. There's some bacteria which produce lactic acid from pyruvic acid. Even in the muscles, when we do extensive exercise or excessive exercise, because of that, there's an inadequate okay. So when we do excessive exercise, there's a lack of oxygen occur in our muscles, and because of lack of oxygen, the pyruvate get converted into lactic acid. Moving next is the aerobic respiration. That's a complete oxidation. And this process takes place in the presence of oxygen. Aerobic respiration in eukaryotes occur in mitochondria. Remember, in eukaryotes, the aerobic respiration occur in mitochondria. And it is represented by the complete oxidation of the organic compounds. First, when glycolysis happens, glucose is converted to pyruvate, right? This pyruvate will move to mitochondria. In mitochondria, this pyruvate will be converted into acetyl-CoA. Okay, and the enzyme involved is pyruvate dehydrogenase. And this enzyme is a very complex enzyme. Why it is complex? But it consists of many units. There's E1, we represent it as E1, E2, and E3. So E1, these NADH, TPP, lipolate, FAD, these all are cofactors, okay? And we know what are cofactors. They are the non-protein part, okay? The apoenzyme binds with the cofactor, then it becomes a holoenzyme, the biologically active enzyme. So all these are cofactors. For E1, the cofactor is TPP. For E2, the cofactor is lipolate. And for E3, the cofactor is FAD and NAD+. And when this pyruvate is converted to acetyl-CoA, NADH is forming and CO2 is released. So it is also a decarboxylation process because CO2 is releasing and NADH is forming. Moving next is the Krebs cycle. Once this acetyl-CoA is formed, it will enter into the Krebs cycle. We also call it as TCA cycle. And this Krebs cycle, it takes place in the matrix of mitochondria. Okay, here's the acetyl-CoA. First acetyl-CoA binds with oxaloacetate. The condensation, this step is known as condensation. And this step will result in the formation of citrate. So citrate is going to form, and the enzyme involved is citrate synthase. Later, this coenzyme will be released. Now with citrate, we'll convert it into isocitrate. The enzyme involved is aconitase. This isocitrate will be converted into alpha ketoglutarate. In this step, NADH is forming and CO2 is released. And the enzyme involved is isocitrate dehydrogenase. This step is very important. This step is an oxidative decarboxylation. Because CO2 is releasing decarboxylation. In this step, NADH formation is taking place. Remember this. All the steps where NADH formation is taking place, or ATP or GTP formation, or here in the Krebs cycle, you can see that FADH2 is also forming. Remember these steps, they are very important. Because generally the question comes from these steps. Then alpha ketoglutarate is converted to succinyl-CoA. Enzyme involved is alpha ketoglutarate dehydrogenase. In this step also NADH is forming and CO2 is releasing. Okay, so this is also oxidative decarboxylation step. Next one, succinyl-CoA is converted to succinate. And the enzyme involved is succinyl-CoA synthetase. This step is also known as substrate level phosphorylation. Because in this step, the ATP formation is taking place. So succinyl-CoA beta, it is an isozyme. And isozyme are those enzymes which have a different sequence, but they perform the same function. That means they are catalyzing the same reaction. So succinyl-CoA synthetase is also isozyme. One is specific for GDP and one is specific for ADP. So what happens? First, GTP will be synthesized, and this GTP will donate or transfer its terminal phosphoryl group to ADP. So ADP will be converted to ATP, and this is a reversible reaction. This ATP and GTP is this GTP and ATP is a reversible reaction. Remember this. Now succinate will convert into fumarate. The enzyme involved is succinic dehydrogenase. And here the FADH2 is forming. Okay, here FADH2 is forming. In this step 1 and this 2, the NADH is forming. And at this step, ATP formation is taking place. So substrate level phosphorylation. This step is substrate level phosphorylation. Remember these steps. And you also know that succinate, its competitive inhibitor is malonate. Remember, the malonate is a competitive inhibitor of succinate, and competitive inhibitors are those which have a similar structure, same as the substrate. So there's a competition between the inhibitor and the substrate for the active site of the enzyme. So malonate has a similar structure with same as succinate, which is the substrate. So malonate is the competitive inhibitor of substrate, and the enzyme which malonate inhibit is succinic dehydrogenase. Okay. So succinate is converted to fumarate. Now this fumarate will convert it into malate, and the enzyme involved is fumarase. And in this process, the water is utilized, so it's a hydration process. Now this malate is converted back to oxaloacetate. Enzyme involved is malate dehydrogenase. In this step also NADH is forming. Okay, in this step also NADH is forming. So total 1, 2, and 3. 3 NADH are forming. 1 FADH2 is forming. 1 and 2. 2 CO2 are releasing, right? And 1 GTP is forming. Okay, these are the end product of the Krebs cycle. But as you know that when glucose is broken down, or when glycolysis takes place, glucose is converted to 2 molecules of pyruvate. Okay, so when 2 molecules of pyruvate will converted, when this reaction of pyruvate, when pyruvate is converted to acetyl-CoA, this reaction will be catalyzed by pyruvate dehydrogenase, right? So when glucose, let me write it down. When glucose is converted to 2 molecules of pyruvate, we are getting from 1 molecule of glucose. Okay, so end product of glycolysis is 2 molecules of pyruvate. So similarly, these 2 molecules of pyruvate will be converted into 2 molecules of acetyl-CoA. Okay, and this cycle, this is only for 1 molecule of acetyl-CoA. 1. Okay, so there has to be a 2 Krebs cycle should be run, because 2 molecules of pyruvate will form 2 molecules of acetyl-CoA. So you have to multiply this reaction by 2. Okay, I hope it is clear now. Moving next is the electron transport chain. Very important. So electron transport chain, it takes place in the inner membrane of mitochondria. And there are 4 complexes are involved. Complex 1, this is complex 2, complex 3, and complex 4. And this is ATP synthase. Sometimes we also called ATP synthase as complex 5th also. Okay. So complex 1 is NADH dehydrogenase. So this ETS will utilize the product of Krebs cycle, which is NADH plus FADH2. Remember, these two are the product of the Krebs cycle. CO2 is also formed there, GTP is also formed there, but this NADH and FADH2, this will move into the ETS, which is the electron transport chain. So first complex is the NADH, dehydrogenase is complex number 1. At this point, the NADH will be oxidized to NAD plus and the electron will be released. This electron from complex 1 will transfer to ubiquinone. Okay, this is ubiquinone. So from complex 1, the electron is transferred to ubiquinone. And in return, 4 protons are transferred from the matrix to the intermembrane space of mitochondria. Remember, mitochondria is a double membrane organelle. It has outer membrane and the inner membrane. So when the electron from NADH, when this NADH get oxidized, the electron is released. This electron from the complex 1 will transfer to ubiquinone and 4 protons are transferred from the matrix to the intermembrane space. Similarly, when FADH2, this is a complex 2 and complex 2 is succinate dehydrogenase. Okay. So complex 2 will utilize this FADH2 as a source of energy, right? And here the electron will from complex 2 will transfer to ubiquinone. Similarly as complex 1. But in this case, remember, there's no movement of proton. At complex 2, there's no movement of proton. Whereas in complex 1, 4 protons are pumped from the matrix to the intermembrane space. But at complex 2, no movement of proton. So when the complex 2 transfer the electron to ubiquinone, ubiquinone will transfer the electron to complex 3. Similarly, this ubiquinone will also transfer the electron to complex 3, which is cytochrome B6, cytochrome BC1. And in return, when this complex 3 receives the electron, 4 protons again are pumped from the matrix into the intermembrane space. Now this complex 3 will transfer the electron to cytochrome C oxidase, which is complex 4th. Complex 3 will transfer the electron to complex 4th. At this point, the mediator is cytochrome C. This is acting as a mediator. And in return, when this complex 4th receives the electrons, in return, 2 protons are pumped from the matrix into the intermembrane space. 2 protons. At complex 1, it is 4 proton. At complex 3, it is 4 proton. But at complex 2, it is 2 proton. Okay. And the electron from the complex 4th is received by this oxygen. That is why we call that in aerobic respiration, the final electron acceptor is the oxygen. And this oxygen, it will receive the electron, 2 hydrogen from the matrix, and it will reduce to water. Okay. Now this proton gradient has been established. Matrix has low concentration of proton. And intermembrane space has high concentration of proton. Okay, proton gradient has established. And this proton gradient is very important for the synthesis of ATP. Now the proton from the matrix, from the intermembrane space, because in intermembrane space, the concentration proton is high. So the proton from the intermembrane space will move or will pass through this F0 channel, which is a transmembrane channel. When it passes through this F0, this proton gradient gets disturbed. Okay, and there's a release of energy. This energy will be utilized by this F1, which is a catalytic site of ATP synthase enzyme. And this F1 utilize that energy and result in the formation of ATP. This is how the ATP formation is taking place in the electron transport chain or the oxidative phosphorylation in mitochondria. I hope it is clear now. And one more thing, 4 protons, when passed through this F0, F1 ATP synthase, it generates 1 molecule of ATP. So when 4 protons pass, they generate 1 molecule of ATP. So when we have only NADH, then complex 1 is active. This ubiquinone is active, complex 3 is active, and complex 4 is active. Right? So we are getting 4 proton here, 4 here, 4 here. Total, we are getting 10 protons. Right? But when we are using FADH2 as a source of energy, in that case, complex 2 is active. Complex 2, then ubiquinone, complex 3, and complex 4. Then we are getting 4 and 2, that is 6. 6 protons. Right? Because complex 1 is not active when FADH2 is used as a source of energy. And we know that there's no movement of proton at complex 2. So when 4 protons give 1 molecule of ATP, if we divide it 10 with 4, we get 2.5 ATP. Or we approximately write it as 3 ATP. When FADH3, 4 protons, we divide it by 4, we get 1.5 ATP. Or we write it as 2 ATP, approximately, right? So 1 molecule of NADH is giving us 3 molecules of ATP. 1 FADH2 is giving us 2 molecules of ATP. This is what written in the last point. I hope it is clear now. Moving next is F0, F1 ATP synthase. We also call it as complex 5. This is the transmembrane channel. It is embedded in the membrane, inner membrane of mitochondria. And this is the peripheral protein part. This one is the integral protein and this is the peripheral part. And this peripheral part is protrude out in the matrix. And this is the catalytic site of the enzyme. This has the catalytic activity, F1. So when the proton pass through this F0, there's an energy breakdown of proton gradient takes place. And because of that breakdown of the disturbed proton gradient will result in the release of energy. This energy will be utilized by this F1 channel, and there's a conformation changes occurs in the F1. And because of that, ADP plus 1 molecule of inorganic phosphate result in the formation of ATP. So this energy is utilized in the formation of ATP. Moving next is amphibolic pathway. Respiratory pathway, it is an amphibolic pathway. Amphi means both. So this catabolism is taking place plus anabolism. Catabolism, why? Because breakdown of molecules are taking place. Like a larger molecule, macromolecules, they are broken down. These fat, the carbohydrate, proteins, what are they? They are the biomolecules and they are macromolecules. So when these large molecules, they are broken down into smaller pieces, energy is released. And this is the catabolism. We defined it as catabolism. In anabolism, from the smaller molecule, a large complex is formed. Okay, say this. The fat is converted into fatty acid. It's broke down into fatty acid and glycerol. This fatty acid, when it breaks down, it produces acetyl-CoA, right? But if we need the formation of fatty acid, there's a fatty acid synthesis should takes place. In that case, the acetyl-CoA can be withdrawn from this respiratory pathway and can be used in the formation of fatty acid. So this acetyl-CoA can be used in the synthesis of fatty acids. So synthesis process is also taking place, and anabolism means synthesis. Similarly, in Krebs cycle, there are many intermediates are formed in the Krebs cycle, like alpha ketoglutarate. This alpha ketoglutarate is involved in the synthesis of glutamate, protein synthesis. Okay. So synthesis is also taking place, and breakdown, that is catabolism, is also taking place. So both the process are taking place in the respiratory pathway. That is why we call that respiratory pathway is the amphibolic pathway. That is both catabolism and anabolism. Okay. It is not only the catabolism that is taking place in the respiratory pathway. It is both anabolism and catabolism. That is why we call respiratory pathway is a amphibolic pathway. Remember this point. Moving next is the respiratory quotient. Respiratory quotient is defined as the ratio or volume of CO2 evolved upon the volume of oxygen consumed, the amount of oxygen consumed. So generally the carbohydrate, they undergo complete oxidation. As you can see in this equation, there's a 6 carbon dioxide is evolved and 6 oxygen are consumed. So respiratory quotient is 1. There's a complete oxidation is taking place. So fatty acid or fats, they has a respiratory quotient less than 1. That's, for example, in this equation, tripalmitin, the RQ is 0.7. And the protein, they has a respiratory quotient which is 0.9. So fat and proteins, they have RQ less than 1. But organic acid, they have RQ more than 1. Remember this. And carbohydrate, they have RQ equals to 1 because they undergo complete oxidation. Organic acid, they have RQ more than 1. Organic acid like malic acid, oxalic acid. Malic acid have a RQ of 1.33. Why they have more? Because they itself has maximum oxygen. So they require very less oxygen for oxidation. That is why if the denominator is oxygen, so if the oxygen is less, then the RQ value will be more. Now, an organic acid, they require very less oxygen because they itself have more oxygen. उनके पास खुद ही इतना ऑक्सीजन होता है कि उनको बाहर से बहुत कम ऑक्सीजन की रिक्वायरमेंट चाहिए होती है, इसलिए उनका RQ more than 1 होता है. Moving next is the question. I hope it is clear to all. First question says the number of substrate level phosphorylation in 1 turn of citric acid cycle. That asking about in 1 turn. Okay. So the number of substrate level phosphorylation in 1 turn of citric acid is 1. Option 1 says 3, option 2 says 0, option 3 says 1, and option 4 says 2. What do you think will be the correct option for this question? Krebs cycle, which is taking place in the mitochondria. They're asking about this Krebs cycle or the citric acid cycle. We also call it as TCA. What do you think? Substrate level phosphorylation. Try to remember the end product. How many ATP or GTP are forming in Krebs cycle? That is why I was telling you, please remember these steps where the ATP formation, substrate level phosphorylation is taking place, or the step where the NADH formation is taking place, or the step where the ATP are utilized. Remember all these steps because they are very important. Priyadarshini, very good, beta. It's 1. If 1 GTP is forming, that means that process is taken place only once in the cycle, right? So the correct option is 3, 1. I hope it is clear now. So when succinyl converted to succinate, this step this is the step where 1 GTP is released. This step is the substrate level phosphorylation. Remember this, beta. Moving next is which of these statement is incorrect. They're asking about the incorrect statement. Enzyme of TCA cycle are present in mitochondrial matrix. Second says glycolysis occur in cytosol. Option 3 says glycolysis operates as long as it is supplied with NAD that can pick up hydrogen atom. Oxidative phosphorylation takes place in outer mitochondrial membrane. Which is the incorrect statement? They're asking about the incorrect statement. Fourth. Very good, beta. Very good, Priyadarshini and Chinchini. Very good. It is 4th. Option 1 says that enzyme of TCA cycle are present in mitochondrial matrix. Yes, Krebs cycle is taking place in the matrix of mitochondria. So all the enzymes which are involved in the Krebs cycle or the TCA cycle will be present in the matrix of mitochondria. So option 1 is correct. Glycolysis occur in cytosol. Yes, glycolysis occur in cytosol in all living organism. Remember this. Glycolysis operate as long it is supplied with NAD that can pick up the hydrogen atom. Yes, because in glycolysis, this NAD will be reduced to NADH. For the continuation of the glycolysis cycle, we require NAD. Fourth one says that oxidative phosphorylation takes place in outer mitochondrial membrane. No, I told you it is not outer, it is inner mitochondrial membrane. The oxidative phosphorylation, that is ETS, electron transport chain. It takes place in the inner mitochondrial membrane, not outer. Okay, so they're asking the incorrect statement. So the correct option is 4. Moving next. Which statement is wrong for Krebs cycle? There's one point in the cycle where FAD plus is reduced to FADH2. During conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. Third says cycle starts with condensation of acetyl group, that is acetyl-CoA, with pyruvic acid to yield citric acid. Fourth says there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. What do you think? What will be the correct option? They're asking the wrong statement. Okay. What do you think? What will be the correct option for this question? Beta, try to recall the Krebs cycle. Remember the end products of the Krebs cycle. FADH2 is there, NADH is there, ATP is there. No, Chichni beta. See, there is one point in the cycle where FAD plus is reduced to FADH2. Yes. FAD is reduced to FADH2. Number 2 says during conversion of succinyl-CoA succinic acid, a molecule of GTP is synthesized. Yes. GTP is synthesized. And this step is known as substrate level phosphorylation. Number 3 says the cycle starts with condensation of acetyl-CoA. Yes, it starts with condensation of acetyl-CoA. With pyruvic acid? No, beta, not with pyruvic acid. It's with oxaloacetic acid or oxaloacetate to yield citric acid. So this is a wrong and the correct option with respect to question because they're asking the wrong statement. Option 4 says there are 3 points in the cycle where NAD plus is reduced to NADH. Yes, beta, it's true. That is why we get 3 NADH. And in Krebs cycle, we also get 1 FADH2. I hope it is clear now. Moving next is which amount of energy is released from glucose? What amount of energy is released from glucose during lactic acid fermentation? More than 18%, about 10%, less than 7%, approximately 15%. It's a very easy question. A direct question from NCERT. Very easy. In fermentation, very less amount of energy is released. Very less amount of energy. Only 2 ATP. So what amount of energy released from glucose during lactic acid fermentation is less than 7%? It's a direct question from NCERT. Moving next, in which one of the following processes CO2 is not released? In which one of the following process CO2 is not released? Number 1 says aerobic respiration in plants. Number 2 says aerobic respiration in animals. Number 3 says alcoholic fermentation. And 4th says lactate fermentation. Priyadarshini says 4. Okay, very good, Priyadarshini. 4th, lactate fermentation. Aerobic respiration in plants, CO2 is released. When complete oxidation takes place, CO2 and water are released, right? Number 2, aerobic respiration animals, yes, CO2 release. Alcoholic fermentation, yes, CO2 is released. But in lactate fermentation, CO2 is not released. Moving next, in question number 6 says, in glycolysis, ATP is synthesized during conversion of glucose to glucose 6-phosphate. Second says fructose 6-phosphate to fructose 1,6-bisphosphate. Number 3, 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid. Fourth says both 2 and 3. ATP is synthesized. They're asking about the station. It says 4. They're asking about the ATP is synthesized, beta. Synthesized. Okay. So when glucose is converted to glucose 6-phosphate, that means the phosphate is added at the 6th position of glucose, right? So in this step, ATP will be utilized, not formed. Second says fructose 6-phosphate to fructose 1,6-bisphosphate. Now, fructose 6-phosphate at 6th position of fructose, there's a phosphate group is present. But now this fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Now the fructose is also added at one position of the fructose also. This phosphate is added at one position. So it is now it becomes fructose 1,6-bisphosphate. So in this process, ATP is utilized. ओके फॉस्फेट कैसे ऐड होगा एटीपी ही तो डोनर है ना फॉस्फेट का एटीपी फॉस्फेट देगा जब जाके वो ग्लूकोज से बाइंड ज्वाइन करेगा उससे और तभी तो हमें ग्लूकोज 6-फॉस्फेट मिलेगा ठीक है ऑप्शन नंबर 3 says 1,3-bis-phosphoglyceric acid to 3-phosphoglyceric acid in option 3 see 1,3-bis-phosphoglyceric acid at 2 position there is a phosphate group is present at 2 position okay so when this 1,3-bis-phosphoglyceric acid is converted to 3-phosphoglyceric acid ATP is released or this 1,3-bis-phosphoglyceric acid will donate its phosphate group to the ADP and the ADP plus the inorganic phosphate will form the ATP. So there's ATP generation. ATP is synthesized in this process. In this process, ATP is utilized. Similarly, here also ATP is utilized. But here ATP is synthesized. That is why the question is asking the ATP is synthesized. So it is option number 3. Now it is clear. Okay, beta. Now it's already 1. Thank you all for your participation. I hope this lecture was informative for you. Thank you all. Have a nice day and see you in next class.